help us to sequence and analyze sequence data from repertoires, as well as many different additions, developments, and new features to some of our favorite packages, libraries, and otherwise. In the sessions that follow here, this and the next session this afternoon, we'll get to see a highlight reel of those tools and the changes which had been made in the last year. In this session in particular, we'll be running through at lightning speed a series of 11 quick lightning demos, which you can see the agenda for here. So without further ado, let's have our first presenter up. We have Duvy and Andy with the ACE configurator for ELISbot. Please give them welcome. This technical demonstration series. So we'll continue on with the agenda and loop back at the end of this session before the break to see if that problem has been resolved. But next on our agenda, please welcome Max presenting ImmuneWatch DETECT.
All right, so my name is Max. I'm from ImmuneWatch, and today I'm doing the lightning demo on ImmuneWatch DETECT. So what do we have here? We have a TCR repertoire, and as always, it's a varied repertoire, many different clonotypes, and a fundamental question arises: what are these T cells recognizing? What is their specificity? Now, we have developed a software solution that is called ImmuneWatch DETECT that answers exactly this. So for each TCR, it predicts the target of the TCR, and it can process an entire repertoire in just a couple of minutes. For example, here in the middle, we have a TCR that is predicted to target an epitope from MART1, while on the top left, we have some TCRs that are likely to bind COVID. Okay, so what type of input do we need to make these predictions? Now, naturally, we support the AIRRC formats, and we only need a couple of columns to get started. So junction AA, vcol, and jcol. Now that we have our inputs, how do we actually make the predictions? So DETECT is a command line tool. It can be run on the terminal, and it's easily installed with a single line command. So let's start making predictions by calling the ImWDetect program. We supply our input repertoire. We specify the output file name. And then very importantly, we also specify which database we're going to use as training data. Now this has a very large impact on the predictions, as DETECT is a scene epitope model. Here we are using the ImmuneWatch database, which is one of the largest ones available, that covers more than 1,700 epitopes. But you can also use popular publicly available databases like the VDJDB or your own custom database. Finally, we enter a license key. And then we can start running the program. And at first it is going to perform some checks, but then it will start making predictions by showing a progress bar. And here we are predicting the targets of 5,000 TCRs, and we just finished in 7 seconds. So we've made the predictions. How does the outputs look like? So for each TCR, we report the epitope target for the TCR together with a score that is the probability of binding. Now we also show both the antigen and the species. So for example, the first TCR is binding an influenza epitope. Lastly, we also supply some sort of explainability. So DETECT explains each prediction by providing a list of reference TCRs. For the first TCR, we see its junction A on the left, and then we see its reported epitope target GIL. Now, for this TCR, we have a list of one reference TCR, where we can see that it is extremely similar to its junction AA, and this is reported to be experimentally confirmed in the reported paper that is also attached. Okay, so we've seen what the tool can do, but how can we actually use it to solve a research question? Now, we're using data from this fantastic paper where they sequenced TCR repertoires before and after COVID vaccination, and they also stimulated with the spike antigen. Now we ask ourselves, can we actually determine which epitopes are driving the immune response? So from the DETECT predictions on this data, we visualize the epitopes that are most prominent in the predictions. Here we already see some epitopes popping up that may be drivers in the immune response. However, we have not taken into account yet the score of the predictions. So let's make it a bit more strict and let's increase the stringency here below by setting a cutoff of 0.05. And immediately we see already lots of epitopes disappearing. Now we can increase this a bit more as well until we see two epitopes that are very prominent. So on top we have an epitope TFE, which when we hover, we see that 55 different TCRs are predicted to bind to this epitope, over 35 different samples in the dataset. Now the other epitope, YLQ, there are 11 TCRs that are reported to bind to this epitope over 9 different samples. So these two epitopes may be important in driving the immune response in this dataset. Now we can increase the stringency even more by going all the way to 0.3, where only TFE remains, where we are very sure about these predictions and very sure that this epitope is actually driving the immune response in this dataset. So you can follow the exact steps of this analysis on our documentation page. And now, what about the performance and accuracy of ImmuneWatch DETECT? At the end of last year, we were the #1 model in the publicly available IMREP23 benchmark, and you can find more information about this benchmark through Kaggle. Now, you can get access to ImmuneWatch DETECT through our website. It is free for academics and non-profits, but we also offer a free trial for industry users. Now, finally, because we know you guys love TCR so much, we 3D printed one. It is at our booth, but you can also win it by going to this QR codes and trying to predict the targets of this TCR. So thanks for following, and happy to answer any questions.
Thank you, Max. Just to repeat that for the people on the live stream who may not have heard, throughout the AIRRC meeting, we'll be taking questions online via Slido. You can find the link to Slido in the Whova app. So for our next presentation, we have Thomas presenting on LZGraphs. Please welcome Thomas.
Yeah, so hello everyone, and I'm glad you joined me on this short demo on our latest package, the LZGraphs. It's an implementation of a recent paper we published about a model for TCR beta encoding using lossless string compression. So the main idea behind our model is leveraging the properties of string compression to reduce the size of a T-cell repertoire of CDR3 sequences to the minimum size possible, yet still being able to make direct inference to this compressed state and derive various statistics and features that we can use in downstream analysis and modeling. So the main aim of my presentation today is to get you familiar with some of the functionalities we provide in our library, which is sequence generation inference, generation of new sequences, informative feature vector derivation, information regarding diversity of repertoires, and a new way to measure sequence importance using the centrality measure. And I'll just note that the notebook here will be provided. I'll attach it somehow to like this session, and it provides very detailed and very broken down explanations about everything I'll mention here rather quickly. So the first thing I would like to show you is how such a graph looks like, okay? So once we leverage the decompression algorithm to break down a T-cell sequence for its unique subpatterns, we start aggregating it using the sequential order in which those subpatterns were detected. So you can see that the first sequence that is inserted into such a graph creates a simple path. But as we start adding more and more sequences, you can quite clearly see there are some subpatterns that are shared between sequences that reappear, and you see them as kind of shared nodes. But interestingly, as we add more and more sequences, you see that the complexity of the graph grows quite exponentially. But don't let it like scare you. It's apparently it's very easy to derive very valuable information from this complex structure. And the first thing I will start is showing you the likelihood that we derive from these graphs. So to simply put it, we can take a sequence, decompose it, and follow the path through a given graph and get the likelihood of seeing such a sequence with respect to a graph. So having a repertoire, we can know, or any set of sequences for that matter, right? We can know what's the likelihood of seeing such a sequence given that set of sequences. And a small case study to kind of provide the fact that the different graphs affect the likelihoods we get. I'm showing right here where we take the source repertoire you see here, which is like the repertoire of interest for us, and we take another data set of repertoires, and we calculate the likelihoods of all our repertoire of interest with respect to our entire data set of repertoires. And then this likelihood fingerprint can be projected onto 2 dimensions. And you see that although those are the same sequences, depending on the repertoire, like the LZGraph used to infer those likelihoods, we get a wide distribution depending on the dynamics inside each of the repertoires. The next thing, okay, and it's like 2 things together, right? So it's both how we can simulate new sequences and how we can effectively use the graph structure to create features that we can then use in downstream like machine learning and analysis tasks. So to generate new sequences, it's kind of intuitive, right? We can take one of our initial states, start making a random walk based on the likelihoods on the edges up until we reach a terminal state. And the resulting path we take results in a sequence. But how can I prove you that those sequences have like the same statistical properties as the source repertoire we used? So that's exactly kind of the case study I'm showing you here. So what we did was basically take the entire set of repertoires we have, selecting randomly one of them and generating a synthetic repertoire based on that randomly selected one. And then using our method of feature extraction, right, using the LZGraph, we project those features onto 2 dimensions again to show you how both like our feature encapsulate the dynamics in the repertoire and both how the generated sequences we synthesized are more closer to the source, the origin. So each point here is a repertoire in our example data set, and you see the red dot is the source repertoire we chose to generate the sequences from. And you see that once we encoded it with the unique features we use in our graphs, right, projecting those features, like using this feature vector, resulted in a distribution that keeps the similarity quite close to repertoires that share the same structures and same patterns. So the blue one, the generated one, the synthetic one, is really close to like the source one, right? Because like we used it to generate it. But other repertoires in our data sets are quite far apart, even though you can see there are some similar ones. So the last 2 things, and like I'll only mention them, but I do encourage you to go to the notebook and try it out by yourself, just swap it with your own data set, are 2 features that I find very interesting and very useful in my day-to-day analysis. So the one is the K1000 index, which is basically a way for us to look at a set of repertoires and ask, what's the most diverse repertoire? We are used to use like alpha and beta diversities, hill-based diversity matrix, entropy-based diversity matrix, but none of them actually formulate and capture the information structure, right, in the information theory sense of the sequences. So K1000 gives us a solution to diversity which is not abundance-based, which is not biased towards like depth or occurrence of certain sequences. So after using the K1000 index and sorting my repertoires, I can focus on one repertoire, let's say a very diverse one. And then the LZ centrality comes in, and it comes to answer a simple question, right? Given a repertoire of interest, how can I detect a certain set of sequences that are the most interest to me, right? And it does that by taking taking a graph. You can see that the parameters to the function is an LZGraph and a sequence, and it checks how many other sequences or subpatterns are shared with the sequence in question. And the more sequences that resemble and somehow interconnect with that sequence, the higher the centrality value will be, basically meaning that sequence has a higher importance in the context of that repertoire. So yeah, again, this notebook should be available to all of you, and I do encourage you to go up, pip install it, exchange the sample data set here and try it out yourself. I really appreciate your time, and thank you.
All right, so we've got a couple of questions for you. Number one, how is this from colored De Bruijn graphs, or rather, what advantage does this offer over them?
Over what? Can you?
Colored De Bruijn graphs?
I'm not familiar with the color and the programs, I'm sorry.
Okay. And then the second question is, have you tested whether the K1000 index is indicative of immune state, such as health or disease?
Well, yeah, like I urge you to like look at the paper. In the paper, we showed how different pathologies, like different states could be detected using like the K1000 index and the different centrality. We actually show how genders can be detected using the K1000 centralities, where in some repertoires and in some conditions, certain repertoires have higher diversity than the opposite pathology or the opposite sex, for that matter.
Thank you, Thomas.
Thank you so much.
Next up, presenting AbSolution, we have Rodrigo.
So hi everyone, I'm Rodrigo García. I will talk today about AbSolution, which is an interactive tool for analysis and exploration of FAB sequence features. Everything starts with a story, and in this case, we all are here because we love AIRRC data, but there are so many big data sets, and these data sets, they always have so much information that you can explore, and you can get lost, and you can everything is mixed, everything is multi-level. You have sequence level, you have sub-clones, you have clones, everything is interconnected. And there are so many different scenarios. In some cases you want to do one type of analysis, in other cases you want to explore other things. So that's why we work on a strategy to make it easy for everybody and to make it also entertaining. So we have worked on AbSolution, which is a software that works with total data, so you can work with as much data sets as you want. You can also work with as many features as you want, because it doesn't run on RAM memory, it runs on hard drive memory. It's also interactive. It's an AIRRC7 application, so you can explore and you can play with it. And precisely because it's an AIRRC7 application, you can try different parameters. It's very flexible. So this is the solution we offer. As I said, it is scalable, works with memory mapping techniques, uses, where possible, linear algorithms. It's an AIRRC7 application, and it's very flexible. It lets you play in many different scenarios, try many different ideas, and you just need to have some time and a coffee, and that's it. So it's divided in 5 steps. The first step is the standard one, file parsing and processing. In the second step, it calculates many features. The third step, you can explore these features.
And then you can filter out some features, and if you're lucky, it will bring you some insight on your data. So it works with PCR or TCR datasets following the AIRRC standard format. It also reconstructs the germline sequence and can work with partial framework region 1 or partial framework region 4. So yeah, it's a standard R-Shiny interface, looks fancy. And the next step is the interesting one. It's where it calculates the features. So it doesn't only infer the germline, but also calculate many features. Features that are related to the repertoire sequence per se, or features that are related to how this repertoire sequence has changed from the germline. And this says something about the trajectory of the sequence. Basically, we have lots of features because every case can be different, and every case can, a different feature can be relevant. There are features that are related to the nucleotide sequence, features that are related to the amino acidic sequence. Some features are only in the raw values, some features are normalized by length, and these features are measured at the whole sequence level, but also at the framework 1 region level, CDR1 level, etc., and so on. And you have these features measured in the repertoire sequence, but also you have the difference between this repertoire sequence and the reconstructed germline. So you know how this has changed. Basically, this table is about 10,000 features per sequence in total, which is a lot. You can easily collapse your RAM memory even if you are working with a really good high-performance computing. But the good thing is that you only need hard drive space. For each AIRRC dataset, it generates this super big feature information table, which is quantitative information. This quantitative information can then be used for a new map or a PCA, and also some extra sequence information that it's not used in the analysis per se, but it's good to have it there in case you want to use it as a covariate or use it as just to color the PCA properly. In this case, this is the easiest example. You can see a mice germinal center dataset, and every dot is a sequence, and this is a PCA of these sequences measured in the germinal center. You can see that the sequences that belong to light chains, they are closer than the sequences that belong to heavy chains. So this works as intended. It's not very it's nothing new, but it's a measure of quality. And here you have these 10,000 features, including a PCA. So you can easily get an overview of your repertoire or repertoires. This can scale up as much as you want. You can add as many data sets as you want. You can color according to different attributes. One of the good things also of this program is that the VJ combination is it's a nice algorithm. So V and J genes of the same family, they always have similar colors. So it makes very easy to explore the data sets. You can also pre-filter features. For example, you can only, I will show it later, but you can only keep features that are related, for example, to dominant and non-dominant clones, remove the rest. And you can use PCA, UMAP, and the good thing about this in Shiny is that you can interact very quickly. You can select, drag, drop, store information, move on, filter. This is really easy to do and really comfortable. For example, you can also explore things at the clonal level. Here you can see the clones that are shared between peripheral blood and lymph nodes. You can select them. You can see how they are shared. You can subset these sequences. Here you can see, for example, sequences with the same V and J usage, and you can see that they are different in dominant and non-dominant clones. So you can also know which features separate those. You can do feature-wise logistic regression to select these features, to separate between the groups that you are interested in, and as you can see, you really separate things. So it's a comfortable, easy-to-do thing, it's interactive, and it's a really good solution. Thanks, everybody. If there is any question, I'll be happy to answer. If not,
I'm just giving it a minute. Write in any questions if they have any questions. Okay, we have a question. Can I upload my own VJ assignments?
I haven't heard anything. Can you repeat?
Yes. Can I upload my own VJ assignments?
Yes. Yes.
Okay.
So it's still an ongoing tool. It has still some work to do, but you can add your own quantitative or qualitative variables. So it's very, the idea is that it's very flexible and you can do anything you want. That's it?
So, round of applause for Rodrigo.
Thanks a lot.
Right, next up we have Vanessa with AnalyzAIRR.
Hello, I'm Vanessa. I'm from the I3 lab in Paris. And today I'm going to present you AnalyzAIRR, which is a user-friendly R package designed to analyze mainly TCR repertoire datasets, but also Ig repertoires. Now, although AnalyzAIRR is developed in R, it is user-friendly for biologists with little or no background in bioinformatics, and it is complemented with a Shiny web application and a guideline that details every analysis proposed with some use cases that can help the user in their analysis. Now, AnalyzAIRR generates ready-to-publish graphics that are completely customizable to the user's liking. AnalyzAIRR takes as inputs either aligned files using MyXCR or any other format that follows the AIRR standards. The user can also supply the package with a metadata file that contains any information that the user will judge necessary for their analysis. AnalyzAIRR will take both of these information and integrate them into a RepSeq experiment that contains the clonotype tables, the metadata, and other information that I will go further into detail in the demo. This RepSeq experiment can be then used to either do a general data exploration of the data set that can help also identify outliers. You can also manipulate the data in order to exclude the outliers that are identified or even sort some particular sequences. And of course, do some comparative analysis to answer a precise biological question. These features will be described further in the demo. For this demo, I'll be using a published data set that contains 18 TCR alpha repertoires of 3 different cell populations: the CD4 effector T cells, naive regulatory T cells, and activated memory regulatory T cells that were collected from the spleen of 6 young healthy B6 mice. I already aligned the files using MyXCR and used the files to generate the RepSeq experiment. I'll be showing you a demo that was done using the Shiny web application, but all of the features can also be found in the source code of the package. Now I'm going to download the RepSeq experiment in here, but you can also upload directly the aligned files, and AnalyzAIRR will generate the RepSeq experiment for you that you can download for future use. Now, when the data is loaded, you have an overview of the metadata, but also some basic statistics, such as the number of sequences and the different samples in the dataset. Now, I will first start by doing an exploratory analysis of my dataset by looking at the diversity using the Rennie index. I will calculate the Rennie index on the amino acid clone level, which in AnalyzAIRR is the V gene, the amino acid CDR3 sequence, and the J gene. I will also color my samples based on their IDs. So in this generated plot, you can see the Rennie profiles, and there are some of the samples that have a steep decline in their diversity at the Simpson index at alpha 2 onwards. Now, if you separate the samples based on the cell subset, you can notice that all of these samples belong to the TF population. Now, to further investigate this decline in diversity in the TF population, I will look at the detailed clone count table at the amino acid clone level. In this table, if we sorted based on one TF sample, like this one in here, you can see that there is one particular sequence that is highly expressed in all of the TF samples, and it is present at counts that are nearly 30-fold higher than the second most frequent clone in the sample. Now, as we're looking at the homeostatic repertoire in healthy mice, we do not expect to see such monoclonal expansions. So I will choose for this analysis to exclude this sequence from the data set. And this is possible using AnalyzAIRR. We can filter out either entire repertoires or specific sequences. So here I'm gonna only filter the sequence out from the TF samples. And when the filtering is applied, there's a history table that registers all of the actions that were performed on the RepSeq experiment, such as here the filtering of this sequence. Now that we've removed any uncertainty in the data set, we can jump into the comparative analysis. We're going to look again at the repertoire diversity, this time only by looking at the Simpson index to compare the diversity of the different cell subsets. We can see that the naive Tregs have the highest diverse repertoire, whereas the AM Tregs have the least diverse repertoire, which is quite expected knowing their phenotype. We can also confirm these differences with a statistical test. We can also compare or look at the repertoire composition of the different cell subsets to compare the repertoire conversions. I'm gonna look at the similarity on a heat map that shows the Morissita scores calculated on the amino acid scores level, sorry. So the Morissita index takes into account both the presence, absence of clones, but also the occurrence of these clones. I will also apply clustering on the heat map using the word D2 distance. And as we can see, this clustering clearly discriminates between the three cell subsets and separates the Tregs in orange and blue in two separate clusters. AM Tregs, interestingly, have really private repertoires, which is not the case for the naive Tregs. We can zoom in on a specific set of samples, here the AM Treg cluster, and look at the Morissita scores that we can also download either to do another representation or to perform other types of analysis. Now, these are some of the features that we propose with AnalyzAIRR, but there are also other features that you can discover. At the end of the analysis, a report is generated with all the plots that were generated during the session. The plots can be commented in the text box and downloaded in different formats. So if you've liked this demo and you're interested in using AnalyzAIRR, you can find the Shiny Web application and the source code on these links. If you have a small data set that you wish to use to test the Shiny Web application, it's also hosted on the Shiny Apps servers, but there are not many resources on there, so maybe if you have a little data set, this will work. Thank you.
All right, any questions for Vanessa? What are the main differences between AnalyzAIRR and the Mixer platform?
Well, we do not do any alignment on the samples. Input files have to be previously aligned in whatever the tool, and we only do the analysis part.
Where do you see your tool going? Like, what's the next step for you? What's the next step for your tool? What do you want to work on next?
I would really like to integrate single cell too, because for the moment, we only analyze or support bulk repertoires.
Okay. All right, round of applause for Vanessa.
Thank you.
All right, just before we head into our break, we're going to take one more crack at the ACE configurator for ELISpot with Duvi and Andy.
Oh. All right. Thank you guys for your patience. We're going to try take two. Hopefully, you guys will believe us that our software is robust, because completely unprompted, we were able to get this up and running on a new computer less than 20 minutes in the heat of battle. So, without further ado, we're going to talk to you guys today about a little side project we were working on called the ACE configurator for ELISpot. I'm going to start off with a quick question. So how many people in the audience have actually ran an ELISpot before? Okay, got some few hands. Keep your hands up if you run an ELISpot with more than 50 peptides. Okay, all hands down. Okay, not bad then. So for those of you who don't know what an ELISpot is an immunoassay that is essentially able to quantify the antigen-specific immune cell activation, and it's commonly used in a principle called epitope mapping, wherein you pulse specific epitopes, and if you want to assay their immunogenicity, you pulse them in with T cells or B cells, whatever antigen-specific cells you were trying to assay, and you get these formation of neat little spots. In today's world, when we try to do like multiple groups of epitopes, we do typically a pooled ELISpot. And so for your pooled ELISpot, you want to put in around K groups of a particular group size, and you want to repeat this over a particular number of replicates. The idea is to ensure that no peptides are shared between pools over more than one replicate. And this allows you to create a almost linearly independent system that you can easily go and deconvolve which of your peptides were truly immunogenic and what was noise. This principle was known as the non-overlapping design and was recently formulated in 2017. However, the issue is that not all nods are created equal. In fact, this little toy experiment shows that even within the nods 1 through 3, you have various degrees of precision. And if you're a wet lab experimentalist and your mental health depends on the number of ELISpots you're asked to run, this makes all the world of difference for you. And so our goal was to set out to try to optimize for non-overlapping design 3. And so what makes nod 3 different? We figured out that depending on where you place these immunogenic peptides, you're able to tune the precision and the recall of this assay, which presents a little bit of a catch-22, because if you knew the immunogenicity of the peptide, you wouldn't really need the ELISpot in the first place. And so we had to come up with a clever strategy of, instead of inferring the immunogenicity directly, we decided if we could somehow group together similar TCR or similar peptides based on their TCR reactivity, this would optimize for precision and recall, given a sample of TCRs without replacement in the actual assay. Some design considerations that we had to include. Sequence edit distance is not the best way of differentiating between epitopes, and there's a small number of examples per PMHC. And so we put together what we affectionately call the ACE neural engine, live software demo coming soon. Just briefly talking about it, what we did was we fine-tuned ESM2, and we're able to group together peptides that share a same TCR and separate out peptides that share no TCR. And that formed the bulk of our fine-tuned ESM. Here is a little plot to convince you guys that it actually works. As you can see, we have the naive baseline of sequence edit distance. You can see here that we're unable to pull apart the peptides that share a same TCR versus the peptides that don't. The same goes with the ESM2 model that was not fine-tuned using our method. And the last plot shows the fine-tuned ESM model. This is on held-out training examples. And so you can see here that we are able to somewhat pull apart the train and test, the in-group and out-group peptides. We further validated this on real-world examples. Here are two pretty well-known epitope mapping experiments where they used ELISpots. We ran simulations with designs of our own, and in both cases, we were able to not only increase the precision, but decrease the total number of ELISpot pools, all for a happier experimentalist. And with that, I'm going to hand it over to Andy, and he'll give you a live demonstration of how ACE works.
So ACE is available on Windows, Mac, and Linux, so all the major operating systems out there. And it comes with two main functions. One, to design a sequence-aware ELISpot experiment, and two, to deconvolve those immunogenic peptides that you're after. So here I'm just going to show you how you can first generate a configuration. So you come over here, and I'm going to load an example with 25 peptides in this case. And it's important to note that we know the sequence of each one of these peptides so that ACE neural engine can infer which peptide should be clustered together. And in this case, I'm going to pull these into 5 peptides per pull with 3 technical replicates. And there are some additional advanced parameters that you can set to tweak a few things to get a more custom design, but I'm going to stick to what we have on the screen for demonstration purposes. It's just going to take a few. Oh, there you go. So what we have here is a peptide assignment into each well. And ACE neural engine has decided that peptides 1 and 2 in this case have a similarity score of 0.7 and that they should be clustered together into the same well. So going back to that not design 3 that we talked about earlier. And you can save these configuration as a file, as a comma-separated file that you can view and print out at the bench side. And so what you'll do is you'll take that, go perform your pooled ELISpot, and then your plate reader will give you those pooled spot counts. And so with those data, you come back to ACE, and we're going to deconvolve those signals. I'm going to load the same example that you saw earlier, same 25 peptides. And here we have some spot count data from those wells. We're going to sort of sort them to get a more visual representation of what's really going on. And then you can come down here to set a minimum positive spot count for a positive well, and the minimum coverage for a peptide to be considered immunogenic. And then we're going to hit deconvolve. And here we see that peptides 1 and 10 are immunogenic peptide hits. In this case, in both cases, they are confident hits. And ACE will give you hits into 2 categories. One is a confident hit, where we think it is an immunogenic peptide. And then the other is candidate hit, which definitely require a second round of assay to establish their immunogenicity. And you can save these results for your publication. And just like that, so easy. So ACE is available for download, as you saw. And this is our customer support. You can reach us at 1-800-ACE-ELISpot. And thank you very much. We'll open up the floor for any questions.
All right, so we have a question. Can you give some intuition of what the previous pooled approaches often aren't optimal?
That's a great question. So the previous approaches, especially the more recent ones, they do a great job of coming up with a sort of, what they use is called a matrix design. And in that, you ensure the non-overlapping criterion, which is no 2 peptides should occupy the same pool over more than 1 replicate. The only issue they have, and this is literally the only issue, is that they are not sequence aware. And so as we can see that there are multiple equivalent classes of non-overlapping designs, each with different results. All we're trying to do is open up the actual peptide sequence as an axis by which we can further optimize the assay.
Great. And then we have one more question. Did you consider location in wells to avoid overlap contamination?
Can a wet lab translate that for me? Well, what does location in wells mean?
I think it's the location of the well itself. So the current design configuration doesn't actually consider well contamination. That's something that we'd like to further work on. In fact, as we're handing this out to our friend who's actually working on it, there are some additional features that we would like to implement to make it more usable at the bench side. So that's a great suggestion. Thank you.
All right, give it up.
Thanks, everyone. We'll now have a short break. We'll be reconvening sharp at 3:10 with report air. See you then.
Gonna go ahead and get started. Welcome back. So this was just a short break, just so you know, we're gonna be having a proper coffee break in about a little bit at 4:10, so you'll get your coffee, no worries. And then just a quick note to our speakers, if you could look at Victor down here, he's gonna be holding up time cards so that you don't have to worry about running over time. And make sure that you talk into this microphone so that our people on Zoom can hear you. Okay, so up next we have Report Error from Ilan.
Hi everyone. My name is Ilan. I'm a PhD student at García's lab in Barton. And today I'm going to present how we can enhance AIRRC7 data analysis using two tools, both PyPair and Reporter. And when we speak about AIRRC7 analysis, we can divide it into three segments. In the first segment, we have what has come from the experimental side, right? Like how we design our data, how we collect the metadata, what is the sequencing protocol, and so on. The next is pre-processing, meaning once we sequence our data, how we process it, what is the design of the protocol, the primers, how we take the data from the rare reads until we can go into the annotation side. And the last is what we call the analysis, where we have tailored computer methods and we have quality control over our data, and we can dive into the analysis and also get the results. So essentially, these three blocks holds a lot of small challenges, right? Standardizing these protocols and creating a very cohesive pipelines that can be reproduced and reanalyzed again and again. It's quite challenging, especially when there are a lot of tools in the environment and also a lot of different run environments, parameters, and so on. So this led us to think about a few things. How we can enhance reproducibility in the world of AIRRC7. By this, we thought about by forming guidelines. The second thing is a lot of the AIRRC7 data world is going into the world of cohorts, right? Multi-sample, not just one sample. So how we compare this multi-sample analysis. And in the end, we wanted to create accessible and interactive platforms that people can use. So today I'm going to showcase PyPair and how it helps with reproducibility and standardization, and also highlight Reporter and how we can explore multi-sample AIRRC7 data. So first off, PyPair, we just published it. This is a collection of guidelines and recommendation on how we can take our pipelines, even our existing pipelines, and what we can pass them through to make them more reproducible and more accessible for the AIR community. So the idea is to use the very standardized way by either creating your own or using current pipelines, but using it in a way of a pipeline format code. So it can either be using Nextflow or a new developed tool that is called Via Foundry, which is a user accessible tool that allows you to develop a pipeline using the Nextflow framework. So the idea is that people will take like pre-made pipelines, they will run it, they tweak the result, get the result, and they will archive it in GitHub. And in the end, use Zenodo to get a DOI number for each of the pipelines, and then it can be long stored. Taking the Zenodo number, what we hope for and wish for is that people will use it later on in the papers to cite it, and then we can move on to create like this reproducible platform. In our work in PyPair, we developed like a collection of 10 different pipelines, 7 pipelines for pre-processing from raw reads until FASTA files, and from FASTA files toward annotation, including genotype and haplotype processes. All of these pipelines were tested to be reproducible and recreate exactly the results that were gotten from the papers. They were all archived in Zenodo, also in Via Foundry. They're also accessible in Nextflow. And by developing this, we started to think, okay, how can we not only verify and reproduce the analysis, how can we think of taking this multi-sample cohort and doing a more testing analysis, understanding if the data that we got is reliable and creditable. So for that, we wanted to develop Reporter, which is a multi-sample quality exploration platform in China that allows us to receive data sets and then create exploration analysis and compare between the different samples through different metrics. So I'm gonna briefly showcase Reporter. This is just a brief demo site. So currently, Reporter is hosted in Hugging Face platform. It's in Shiny base. This is just a demo, and it's in a beta form that we're gonna continue working on. But the main frame shows you the cohort that we have currently. This data set was developed in Steve Bossinger and Corey Watson lab. This is the data set of macaques. And it currently holds closely to 600 repertoires from closely 200 different monkeys. And we have 3 chains, both the heavy, the light, and both the IgA, the IgK, and the IgL. And what this app allows us to do is to go into the multi-sample report. In this multi-sample report, we can choose from each of the chains. We can either take one or choose all. So for this example, let's take the IgK, and it allows us to select different features. Now, these features were compiled to compare between the different data set and get a metric on the control, on the QC, so we can understand if our experiment worked correctly and if there's any deviation between the different samples. So we can select the features that we want. In this case, we can toggle, let's say, all the pre-processing, and we can also take maybe some data from the FASTQ. And once we selected our feature, we can save it, and then we can select our samples. So either we can select a few samples, or we can even select all. And once we select all, we get this this plot application that shows us all the different features that we selected. From here, we can even highlight one sample and see how it changes through all of the features. And we can export our feature table. So just to summarize everything, so we showed how guidelines for creating reproducible AIRRC data, and we also showed a friendly platform to create multi-sample reports. We are hoping to integrate more and more feature of both sequencing, pre-processing, and analysis step into the report AIRRC for the multi-sample comparison reports. So I just want to acknowledge everyone that were part of this project. So my lab, Boer and Vera, that worked a lot on this project, and William as well. And from Corey, Melissa, and Steve Bossinger lab. And for the RepCred, Arthur, that helped us a lot. So thank you.
So our question is, can you change pre-existing pipelines in VIA Foundry?
Yes, so you can. You can change pre-existing pipelines. We uploaded 10 pipelines to there, including all of the processing steps and the models. And you can take and copy one of the pipelines, modify as you wish, both the parameters, and run in the environment. We're closely related with VIA Foundry company to help the community.
Thank you, Ilan.
Next up, we have Chaim with Alligator.
Nope, that didn't work. Oh, there we go. Okay, great. So this is Alligator, which is our annotator of loci for immunoglobulins and T-cell receptors. So the idea here is that if you get a genomic contig from an Ig or a TR locus, you can put it through Alligator and get back annotations for the V, D, J, and C genes. So the conceptual framework, there are 3 modules in Alligator, 3 commands that it takes: extract, predict, and annotate. And I'll go through each of these. Extract is to extract reference annotations from an IMGT webpage because it works by comparing, by blasting against known genes. Predict is to get the RSS sequences to assess functionality. And then annotate is the actual business module. So for prerequisite installation, the install is just straight cloning the GitHub. Prerequisites are Python 3.6, Muscle, Blast, Bed Tools, and some Python packages that are in requirements.txt. So should be pretty straightforward to get up and running. So the first module or command is extract. So we just, you can see the basic form of the command here, and you can take an IMGT web page in HTML that looks like this with all of the annotations for a reference locus, and get back a nice bed file that has the same annotations that can be used by Alligator. The second module is predict, and this is using the recombination information content to identify likely CSS. And this is done, so this is the original paper that derived the RIC, and then this is a paper that implemented it in a tool called DNAgrep. And so we just have locally cloned DNAgrep, and it goes very quickly just through a whole genomic contig. You can get through a couple megabases pretty quickly and identifies the sites that are likely to be RSS. And then finally, annotate. So this, you give it the contig that you want to annotate. So we've included in the repo this contig from Ramesh et al. 2017, which is a genomic contig from a rhesus macaque. And you can, we've already included the RSS predictions, although you can regenerate them using the command I showed before. And then we are going to tell it we're going to annotate an IGH, although you can also use IGK, IGL, TRA, or TRB. And then this is our reference genome, which is going to be the IMGT annotation of REMAC10. So this is the IGH locus that IMGT is extracted from REMAC10, and then here's the bed file with the IMGT annotations for that were generated with the extract command. And then in addition, you can provide an optional coding DB to compare to help with annotation and naming. And it produces a bunch of output that looks like this. So it identifies possible genes by blast, and it looks for maybe if there's a splice donor in an alignment gap, or if there's thinks there's a pseudogene because of splicing issues or stuff like that. Most of this stuff gets printed to a log file, so that you don't have to have it clutter up your screen if you don't want to. Some naming things. So that this IGHVX here is basically saying that it's more than 25% divergent from all genes in your reference contig, and that typically means it's a pseudogene anyway, but we're going to work on refining that. And then if it matches a known gene either in the reference assembly or in your coding database, it'll take that name, and if it's an exact match to an allele, it'll give it that allele name. And if it's not, then it uses the SHA1 hash, and it takes the first 4 characters of that, the hash of the nucleotide sequence, I should say. And so it just, that way you don't get clashing allele numbers and don't have to worry about database updates and things like that. So it's just based on a hash. And if it's not within about 5, it's not within 5% of any known gene, then you get, I don't have an example here, but you can, with like this, where the hash actually gets used as the gene name instead, and then we just give it a star of 1 allele. And so then it produces a GFF output like this, so your contig, and we annotate, the overall V gene, the RSS V exon, the L part 1 exon, and with the RSS, we can include the RIC score here, positions, all the just basic standard annotations. And again, looking at the naming here. So just a few considerations is right now we're using rhesus macaque reference to annotate cynomologous macaque. We've also looked at using rhesus for human and human for rhesus and human for cyno. So within that relatively close range of divergence, it seems to work pretty well. I don't know yet how that would work if you tried to use a human reference to annotate, say, a mouse contig or something like that, or if you need to do salmon IG. I don't know how well that would work. The other thing that we run into is that the RSS model in RIC is human. RIC also has a mouse RSS model, but it doesn't do any other species. And we think that we're running into issues with the macaques where it doesn't pick up all the RSS the way we would like because of that. And so we're figuring out how to handle that and whether we can maybe rederive an RIC potential. And it's still in beta, so the GitHub is tagged as a beta release, and we invite feedback and ideas for future development. We're going to get it sanded down in the next couple of months and really ready for production. And I want to thank Simone, who's been leading the development of this package, and I will be happy to take questions.
Any questions for Chaim? Okay, thank you.
Right, next we have Steve with incantation.
All right, so I'm going to talk about incantation, a start-to-finish AIRR-compliant analytical ecosystem for AIRR-seq data analysis. Before I jump in, I just wanted to point out Susana Marquez, who's in the back right corner over there. So Susana is the person in the lab who is tasked with making sure that incantation works together as an ecosystem and interfaces with a lot of people using it to make sure we also respond to a lot of user queries on feature requests and just basically how to get started. So my lab has been involved with B-cell receptor sequence analysis since the days when we used to get files with 50 B-cell receptor sequences, and we used to think that was a large data set. So we've been in this business for a while. The incantation suite started out with two toolkits, Presto and Changeo, that were published in 2014 and 2015, respectively. So we are hitting now our 10th anniversary of incantation, which is to say, you know, we've been developing this for a while. It is a very, very stable system at this point with a lot of tutorials. We've worked out a lot of the kinks in the system. So if you're looking for a framework to get started in AIRR-seq data analysis, we highly recommend you take a look at incantation. We were born with a focus on B-cell receptor analysis, so we have a lot of methodology for dealing with somatic hypermutations. But the core methods are also applicable to T-cell receptor analysis, and we have several groups who are using incantation for that as well. We have done a lot of work over the past several years to update everything to single cell, so we are compatible with single cell methods, paired heavy and light chains, as well as integration with single cell RNA-seq data sets that come along. Incantation is a modular ecosystem, so we currently are composed of 7 different core packages that handle different aspects of the functionality, and I'll step through some of those key packages in the later part of this presentation. But just to point out that what makes this all work is the AIR community data standard. So we are fully compliant with AIR community standards, and that's what allows the tools to talk to each other, and that's what allows our tools to also talk with other methods. We are released under AGPL v3, so we are free to use. We are open source and available to both academic and commercial. You can install all the packages individually, or you can install using the Docker container, which includes all of the packages, as well as reference scripts and other libraries that go along with a single Docker pull. We are widely used by the scientific community, so many of you may have seen posters already that use incantation as their core analysis framework. We hit over 500,000 cumulative downloads earlier this year, used across many countries as well. So if you're interested in getting started with this, we also work with a lot of groups that are new to this to help them get set up with their initial pipeline, so please reach out. As I mentioned, we are interoperable. The default file format in incantation has been the AIRR-seq standard as of release 4.0 in 2020. So this lets all of the tools talk together. Importantly, You don't have to fully use the incantation suite for every single step, but our goal really, and this is part of the AIRRC community in developing these standards, the goal is if you have other tools that you want to use for part of it, the diversity analysis or any other aspect, you can plug and play incantation packages with other tools, and they should all be able to talk to each other through the AIRRC community standard. Just a couple slides on some of the what some of the packages do. So the Presto package deals with going from raw reads to error-corrected assembled sequences. We support we have predefined pipelines that support many many sequencing libraries such as NEBNext or the Takara. If you're a company out there and interested in getting your pipeline into incantation, please do reach out to us. For VD8DJ annotation, we support the output of HiVQuest and Ig-Blast, Partis, Cell Ranger, and other tools, and you can run tools like Ig-Blast from directly inside of incantation. We heard earlier yesterday about the importance of germlines. So incantation, we originally developed Tigger, but we also support packages like Piglet to create personalized germline databases and identify novel V genes carried by individuals. Cloning, of course, is an important part of the analysis, so partitioning your sequences into clonal groups. And clones are obviously, you know, they are used as the basis for things like diversity analysis, mutation analysis, reconstructing lineage trees. We've developed a lot of methods and incorporate multiple different approaches to identifying clonally related sequences within incantation and supporting the thresholds for cloning. So it's common, for example, to look at CDR3 regions and use some method of similarity to partition into clones. For example, 90% nucleotide similarity in the CDR3s. But what exactly that threshold should be can depend on the data set. So we support methods like analyzing the distance to nearest graph, which you see on the right, for identifying on a project-by-project basis what the appropriate thresholds ought to be. We also have Shazam, which deals with hot and cold spot modeling. We all know there's intrinsic biases in certain nucleotide motifs that tend to be hit more or less by the somatic hypermutation process. So we have methods in Shazam for building models of which DNA motifs tend to be targeted, and you can compare those across, say, disease and healthy control samples. We also support methods for looking at selection. So it's common to look at compare the replacement and silent mutation frequency to find evidence of selection. If you have more replacements than expected, then that's an evidence of positive selection. So we have the baseline method where you can incorporate and make those inferences about selection that really incorporate background models of hot spots and cold spots, which is really important for doing those kinds of calculations. We also support diversity analysis through the Alchemab packages. As we've heard a few times, there's multiple different ways that you can measure diversity. We support this Hill index approach where there's a parameter Q shown on the x-axis here. And depending on what you set that parameter, you have different definitions of diversity. Going down towards Q equals 0, where you have diversity as simply richness, the total number of clones, versus if you make Q all the way on the right, a higher value where you're measuring diversity essentially by the size of the largest clone. And we also support resampling to try and equalize the depth across different samples, which again is a key factor in doing diversity analysis. For lineage tree reconstruction, we have our newest package in the suite Dowser, which is headed now by Ken Hain. And this allows you to take clones and reconstruct lineage trees using multiple methods like maximum likelihood and maximum parsimony. I urge you, there's a tutorial where I think there's maybe 2 or 3 spots left in this afternoon. If you're interested in lineage trees, please do sign up for that tutorial. And finally, while incantation, the suite is really designed for bioinformatics scientists. So its core functionality is really flexible and allows you to develop pipelines for many, many different kinds of experimental designs. As things are becoming more standardized, we recognize there's sort of very common workflows that go through AIRRC data analysis. And we've developed now a workflow written in Nextflow where you can essentially, in one command, you can run an entire AIRRC analysis. And I would there's also a tutorial on that, although I think that one is full, but please do reach out to Giselle or Susanna if you're interested in learning more about Nextflow. So as I mentioned, there's several different incantation sessions here. There's several posters that in the session that just ended, but please do check out our 2 tutorials, one on Dowser for lineage tree building, as well as the airflow, Nextflow workflow. As well as tomorrow, there's 3 posters, one on incantation, a second one on using incantation, applying it to study Lyme disease. And in that case, we're looking at single cell data sets from skin biopsies and from blood in Lyme. As well as the final poster on how you can do genotyping from single-cell RNA-seq data. And finally, we do a lot of outreach, and we really try to respond to inquiries from the community. So please check out our website at imcantation.org. We monitor very closely the imcantation@googlegroups.com email address. So we are very responsive if you have questions about getting started or feature requests. As I mentioned, it's all open source. You can get all of the code on Bitbucket, and reach out if you have any questions. And I will end things there.
Okay, Steve, we've got something that's like half comment, I think. So different apps doing VDJ calling is reminiscent of multiple motif discoveries. Comparison was disappointing. Would it be the same if VDJ callers were compared?
So there's been, I think, a couple of publications. So VDJ callers, I assume this is things like VQuest or Pardis or Mixer and things like that. So there are some good benchmarking works that are out there. So there are clearly differences between those VDJ callers. I think Goriari has a recent either preprint or publication that's going over some of that. So yes, there are differences, and I think you should talk to him if you want to know the latest on the benchmarking.
Okay, thank you, Steve.
Right, and next we have Anne with Mab Factory.
So, hello everyone. So, Mab Factory is a database we have developed and with features we think are essential when you want to develop antibodies, or when you are developing antibodies and you want to know what the others are doing on the same target. So you have some numbers on so there's a demo instance where you can go if you'd like to explore a bit. There are some numbers here. We have about 80,000 antibodies in our database, accounting for about 3,000 different targets. So the principle is quite simple. You have your target here, let's say, for example, team 3. You have different data on the target itself, and things we find quite important, for example, with which other proteins is this protein interacting, or what are the antibodies with INNs already there for this target. And also things you see here, the number of publications for antibodies against this target. So basically, is it rising or going down? So should you go for this target, or is it already old-fashioned? Then you have protein, some details on the protein, and some details we find very important, again, to develop antibodies. So first, the isoforms. You should be very careful on which isoform you target for your antibodies. Also, the natural variants that exist. The post-translational modifications of this target, and what are the available 3D structures for this protein. And also down here you have the orthologs, because very often you would like to have an antibody that is cross-specific with, for example, cyno, so you can test it in animals, and preferably also with mice, would be nice. So you can just select different species there and make your align it and see, sorry, and see where are these proteins different between the different species and where are they similar. Then after that you have what we call competitive landscape, which means who is working on this target in terms of antibodies. So here you have a list, for example, of the companies that have been patenting antibodies against TIM3. You have a small search engine for publications where this is the default query, but you can add your own keywords or remove some and filter the results here. Same thing for clinical trials here. So again, a default query and which you can edit and here you can filter the results. For example, if you want to see only the clinical trials on cancer, or you can sort by year, for example. Then you have the clinical trials by company and by phase. So if you want to know who is where in these clinical trials. You also have a table here of the antibodies that have been used in combination or as comparison with antibodies targeting TIM3, for example. So they are not all against TIM3 because, as you know, combination, you may use two antibodies that have different targets. Then you can know also in which therapeutic area you have, so clinical trials have been conducted. For example, you can see here that there has been different phase 3 trials, but mostly in osteoarthritis and 2 in cancer. Finally, you have the last tab here is antibodies. So here, the rest of the information is mostly aggregation of existing databases. Here, for antibodies, we really read the patents and go to search for the different information that can be found on each antibody. So you can see, for example, we have functional information. You have details here. So, for example, for this antibody, we have in vivo and in vitro information and different measures here of IC50, EC50 for this antibody and for different assays. They are, when available, you have the epitopes, you have the affinities, and some cross-specificity data if they are present in the patent. For example, you can see this antibody binds to the human and the cyno TIM3. And to see the similarity between these different antibodies, you have here tree-like structure where you can see, for example, so here it's a bit small on the screen, but for example, this one is the sabatolimab, which has an INN. You can see there are many different variants of this antibody in different patents. Here again, you can have all the details on each antibody. And there you can select also the source of antibody if you want to see only those coming from patents, only those with affinities, for example. I can go to the tab, or affinities and epitopes. And you can also select by company if you want to see only the Agenus antibodies or only the Xencor antibodies, etc. And that's it. And if you have questions, I'm on my stand upstairs, so you are very welcome. Thank you.
Okay, thank you. So Does the software scrape patient data from both the United States and Europe, and how often is it updated?
Oh, so the software doesn't scrape patents. I scrape patents. No, this is very important because, as you know, with AI methods, and we do develop a lot of AI methods, but not for this, because we want to be really sure about what we put in our database. So this is manual, and it's not only United States and Europe. We have Japanese, Chinese, Korean patents. Any patent that we find useful is there. And also for clinical trials, it's a direct connection with the API of clinicaltrials.gov. So it's up to date. And yeah, we update the antibody data once a week. Thank you.
Okay, finally we have Ilan presenting RepRed.
So, hi again. I felt like you missed me, so I came back. I'm gonna present RepRed. This was a package that was developed in the software working group. RepRed was developed to the aim of creating a package that will allow us to test the credibility of a single repertoire. So again, in the realm of like AIRRC7 data, we have a lot of challenges. A lot of them stems from the different protocols and analysis that is happening to the repertoires. And knowing if the repertoire is reliable and credible to work on for later on analysis is incredibly important. So the software working group wanted to create an application that will allow us to explore repertoires and assess their credibility, the same as FASTQ is doing for the sequencing for the FASTQ files. So RepRed was developed into a simple, easy-to-use user interface that encapsulate many features that allow us to explore the repertoire in question. So the installation of RepRed is quite easy. You can either install it from our GitHub in R, or you can download the Docker image and run it through there. Using RepRed is also a dual method. You can either run it in R and open a user interface, or you can use the command line and run it through Docker, either uploading a user interface or even running directly the report creation. So I'm going to briefly show the user interface. It's quite simple, actually. The user interface allows us to browse and upload our repertoire. It has to be in an AIR format and a TSV file. If it's not in an AIR format, don't worry, the app will tell you that it's not AIR. We can, if the repertoire is quite large, we can even downsample it and only check part of the sequences to save time in this case. We can also upload a germline references that was used to annotate the repertoires. Once we upload the file, we can select an example data file. We can run the analysis. It will take a few seconds to run. On that time, it creates the report for us. We can either open it directly or download it locally. Opening directly will open an HTML file that contains the report itself. The initial part is just telling us what was the input parameter to run this report. And then we go into the quality control statistics. In this page, it summarizes if we have any missing data, if we have any missing columns that are required for later on analysis. And in the sequencing method, sometimes you get nucleotides, sometimes you get letters that are non-nucleotides, so we can also detect that through RepRed. Going further, we compiled a list of statistics that we thought that could help us analyze the repertoire and assess if it's credible to use. So we analyze the amount of productive sequences there is, and we compare productive and non-productive sequences. We look over the sequencing length to see that it's within the region that we desired, that it matches the sequencing protocol that we have, and we get a table of the statistics as well. We can also look at the annotation calls of the alignment to see that we got exactly what we wanted. So if we think that this data set came, for example, from sequencing a lymph node, so maybe there is a clonal expansion, and then we would expect to see a higher usage of one of the G, the V, the D, or the J. And if not, then and if it was sequenced there and we see that everything is distributed equally, that it can indicate that there was maybe a problem within the annotation file. We can also look on the relative usage. And further, we incorporated in RepRed the SumRep package that was previously developed in the software group to get further on statistic on the hotspot within the sequences, on the cold spot, and on the GC contact, the same as we look at the GC contact in the FASTQ files. So just to demonstrate basically what could be a bad repertoire. So just for this example, we uploaded a repertoire and then we get like this immediately warning. We don't have even a sequence alignment here. There is no contact at all for almost all of the columns, meaning there was probably an error analyzing this repertoire. If we go into the statistic, we see that we have some productive sequences, some non-productive sequences, and we can see the division between them. And because we didn't have the sequence alignment column, we cannot obtain the sequencing length. And this should give us a sign of warning that maybe we should further look at prior steps. So this was just a brief demonstration of the user interface that you can use, and it's found in GitHub and in Docker. And lastly, I just want to briefly say that RepRed is going to be integrated into iReceptor. You will be able to choose samples and then run the analysis and directly export the RepRed report. And I just want to say thank you to all the working group, the software working group that helped create this.
Okay, so someone wants to know, how can I get away with a non-AIR compliant file? Is there a minimum set of columns that's needed for running RepRed?
So RepRed was developed in an AIR subgroup of the software group, so it is AIR compliant, and it has to be AIR compliant, so unless it has the AIR columns, like, it would not work.
And then there's another question, will RepRed be supported long-term by the AIR community?
I guess that will be determined.
Fair enough. Okay. What are the plans on how to develop it further to cope with new sequencing technologies and annotations, for example?
So I guess this is open for discussion for the community. If there's more features that people would like to see integrated into this RepRed report, we'll be happy to take notes from everyone and try to incorporate as much as we can.
Thank you.